welcome you to welcome Scott McKinley from Tulane University. Scott has a PhD from Ohio State in 2006, after a postdoc at Duke from 2006 to 2010. And from the beginning, so stochastic polymer and transport of microparticles. And he joined, so first he was assistant professor from 2010, 2015 at the University of Florida, and now is since 2015 associate professor in Tulane. And so, Scott, welcome. Thank you.
All right. Thank you so much. Thank you for the invitation. Again, thank you everybody for being gracious for coming to the Zoom version of the talk. I apologize for not being able to get up there this time, and maybe I wouldn't have been able to either way because my little one came home with the flu a couple weeks ago, and of course she was better in 3 days, but Dad is still trying to come back from this thing. So any mistakes that you see in the slides are a mixture of the fact that this is the first time that I'm presenting a lot of these results. A lot of them were generated by, really driven by a graduate student who's going to be graduating in April. And so I'm excited to share some of these results, and then any other mistakes is due to the cold medicine. But yeah, please interrupt me with questions as we go along. If you want to put something in the chat, just let me know. And like I'll keep my schedule open, tomorrow and Wednesday for anyone who wants to talk individually about anything you are interested in. So this work, this work is joint work with Christine Payne, who is a mechanical engineer at Duke University. A lot of the data that you will see was collected by the graduate student Nathan Rayans. And this is part of a Simons Foundation NSF center called the Southeast Center for Math and Biology. It's headed up at Georgia Tech. And it's winding down right now. A lot of that is being consolidated into the one big institute at Northwestern, and I think starting this year. So the premise of the center was, the director, Christine Heitch, had the idea to build an institute around the idea that we wanted to, directly have an interface, grow the interface between math and biology, where she basically played matchmaker. She found mathematicians from around the Southeast and introduced them to experimentalists. And one of the main rules was that the experimentalists really need to have their own, generate their own data, because the entire idea was to have a collaborative and an iterative process in terms of learning about the biology, improving mathematical tools for studying it, and then iterating to new experiments. And so we did that. Keisha Cook was a postdoc on this, and she's at Clemson now as an assistant professor. Lindo is the graduate student who is graduating this semester, and Riley Juniman is an undergraduate. I'm not sure if I'll be able to talk about her work, but a lot of this goes back to her honors thesis, and she's a PhD student at Stanford right now. So for those of you who aren't familiar, so when we talk about intracellular transport, the main thing to think about is that you have a cell. This darker region here is the nucleus that has lots of materials around it. And if you follow any of these little white streaks that are sort of running around, these are lysosomes, I believe, in a HeLa cell. The types of cells won't necessarily be important here as much as it is that there are geometries that we're thinking about. But what you'll notice is that in contrast to what we all learn about Brownian motion, these cargoes that we track tend to follow along microtubules. They're driven by molecular motors, and they have a tendency to go in short bursts, and then they pause, and then they go in another burst, and they pause again. And when you look at a video like this, you want to ask yourself, how do you summarize the behavior across a cell? And one of the first conversations that Christine and I had, she was talking about, basically she would have treatments. She was interested in the impact of toxic nanoparticles inside of cells and whether or not they interfere with the transport mechanisms. And to the eye, she says sometimes you put in some kind of genetic, knockout, or you do something and you look at it and you say, wow, stuff's not moving very much. And then you have this other cell where stuff seems like it's moving a lot. And so how do you put a number on that? And how do you understand the variation that goes around that number so that you can go towards actually saying that there is a legitimate biological difference between, two different cells? So, the geometry of cells can be, really different. Neurons, you have like the cell body and a long, long axon. You have, these dendrites. There are other cells where maybe there are these these microtubules or these little roads that are all throughout the cell. The important thing to know is that the microtubules have plus ends and minus ends. Sometimes they are really long and they run the length of the cell, and other times they're much shorter. Every different type of cell has a different, type of geometry. Um, and so, what we're usually tracking is the cargo, and the cargo in this case is going to be lysosomes, but it could be organelles, it can be other types of microparticles. Kinesin motors tend to pull the cargo towards the plus end. Dynein motors tend to pull it towards the minus end. Essentially, every statement I might make of this kind has some kind of exception in some cell somewhere, so please keep that in mind. And a traditional way to visualize a lot of movement within the cell are these things called chymographs, where basically what you're seeing is that across left to right, this is space, and then top to bottom is time. And so as a streak, this like white streak right here moving to the left, that's a cargo that's being, moved in the retrograde direction, maybe towards the minus end of the microtubule. But then there are others that go in the positive direction. And you can notice that there's like a roughly constant speed. So this one's moving fast, but then it starts to slow down and then it starts again. So it's different velocities and pauses and different behaviors. You're trying to capture all of that. So the traditional, way to characterize particle movement is through a quantity called mean square displacement. And so the idea, the motivation for mean square displacement is pretty straightforward. A lot of these processes we might expect that they have, especially if it's, Brownian motion, or if it's something where you're not sure which way it's going to go, it might have mean 0. And so if you're expecting the mean to be 0, then you would look at the variance as the next thing. And for Brownian motion, the variance grows linearly over time. So essentially what you're trying to do is say, what's the growth of the variance of these particles? And, for, like I said, for normal diffusion, that grows linearly. And so, this is a popular enough method. I pulled this image from Wikipedia, so that it must be state of the art if it's on Wikipedia. So normal diffusion, you have a linear growth of the mean square displacement over time, and the coefficient called the diffusivity is the slope of that line. And then there are other situations where if it's sublinear, we might call it subdiffusion, or if it's super linear, we call it superdiffusion. And if you just give it a quick moment's thought, if you have a constant velocity, then you're going to the square of your distance traveled is going to grow quadratically. And so active transport, will grow like with a slope on a, notice this is a log plot, slope. Well, I'm sorry, this is not log, but this will grow quadratically. On the other hand, if you have an anchored diffusion, this means that something is stuck and then it fluctuates around that point. There'll be an initial phase where it starts to move out to its sort of radius of fluctuations, but then it'll sort of stay there. So the variance will sort of reach a saturation point. We might call that subdiffusive, maybe stationary, an anchored diffusion or something like that. If it hits an actual plateau, it's anchored. If it goes up slowly, it's called subdiffusion. And the key thing to understand is that if you're trying to diagnose or understand like a whole bunch of particles and their behaviors, the mean square displacement does not uniquely describe what's going on. So multiple stochastic models can yield the same MSD shape. So for example, for a linear mean square displacement, yes, Brownian motion can do that. But also, if you have a particle that's switching between being stuck and then diffusing, and you zoom out and watch it for long enough, that will eventually have a linear mean square displacement. And for sublinear mean squared displacements, you have fractional Brownian motion. Generalized Langevin equation is something that I've studied for quite a while. And when my original work was mostly studying diffusion in mucus, healthy mucus and unhealthy mucus, and the generalized Langevin equation played an important role there. And then there's also switching between Brownian and anchored diffusion. So if you're anchored for a really, really long time, so you get stuck and you stay there, they call this like caging effects, and then you move for a little bit and then you get caged again, you can get a sublinear mean square displacement. So the point is that just plotting a mean square displacement is not sufficient to tell you what's going on. Nevertheless, it's often a good diagnostic to start the conversation. So for the types of particles that Christine Payne is interested in, these lysosomes, she's interested in the impact of the size of the lysosomes. So basically, they're the garbage cans of the cell. The original motivation for this project was she was interested in titanium dioxide, which are these large particles. They have a tendency to swell the lysosomes. And the question is whether or not the enlargement of the lysosomes affected their transport overall. And so she was looking in particular at contrasts, larger and smaller cells, but then the paranuclear region, the region near the nucleus, versus the periphery of the cells. And so if you just take the individual particles and you track them, and then you do this mean squared displacement, and you average over all the particles in the periphery and all the particles in the paranuclear region, you get these two different curves. And you can see, hey, there's more activity happening in the periphery than there is in the paranuclear region. So, okay, there's something to study. So, what can we say about this? Well, you can do a common tool is called pathwise mean square displacement. And the idea here is that you go path by path. And for every path, it's like a sliding window average. You look at increments of a given time lag, you square that, and then you sort of average over all the possible windows. And so you get an idea from, some paths will be stuck and some paths will be moving. So what I did in this particular plot is any particle that had a square displacement that reached a certain level, I think it was 0.01 micron squared, I would just declare that not active because it never moves. So it's, so that's in pink. And then the gray ones are paths that did eventually move. And you can see that there's a clear distinction. You can put a threshold and sort of separate them like this. You can label them. And you can start to get a clue that in the paranuclear region, there are simply more stuck particles. The proportion of particles that are stuck is much larger. But of course, everything about this is sort of everything about this is sort of ad hoc. Like I set that particular threshold, but why is that a good threshold? What happens if I pick other thresholds and so forth? So what you want to start to do is be able to characterize these things in sort of a way that is going to be somewhat universal so that anybody can use these statistics. But then also, even though I know they're more stuck or less stuck, I still don't know, well, is it that they're moving slower? I mean, maybe they're active, but they are just moving slower than they were otherwise. Or is it they're genuinely immobilized for longer periods of time? And so that part right there starts to introduce the idea that we really want to break these paths down into their different segments, because the mean square displacement, by doing that sliding window, intrinsically, if you have a multi-state process, one that's paused for a little bit and then moving for a little bit, you're going to be averaging over the pause state and the moving state in a way that's going to be hard to disentangle those. So at some point, you sort of want to say, well, why don't we break these things down and look at them directly? And so, of course, in the literature, there is some, there's a great initial analysis of this kind from Sandra Angelada, and Goldstein's first name is escaping me at the moment. But their basic idea is they were taking these chymographs that I mentioned, and they basically built this tool. It's called a chymo analyzer. And the key here is that it is an automated analysis of user segmented paths. And so the first thing I want to say is that this is a really great, really interesting idea. It's a shift in the way to think about how to analyze these particles, but there are always these interesting choices that come up. The first one, and this is one that goes across particle tracking actually, no matter what you do, is what is the impact of the human hand? So in this case, the chymo analyzer requires someone to sit down and actually say, well, I think that there's definitely some kind of switch that happens here, but is there a switch that happens here or not? Are segments 1 and 2, should they be combined? This is a pause, then it's moving, another pause. A lot of it's in the eye of the beholder, right? So, you know, you could imagine from one person to the next person, they could look at the same path and have two really different interpretations of how many segments there are, you know, how refined we should break these things down. And then, you know, what's worse sometimes is that these things cross. And you're like, okay, well, I'm pretty sure that's the same path, but, you know, I mean, there's a whole host of things that are very difficult about this. So you should always keep that in mind. The second thing is that they have these distribution of velocities, which I found a fascinating concept because if I'm talking about a cargo and I know these things are sort of fluctuating, you'll see paths. I mean, there's no such thing as like a velocity, like you're sitting in your car and you're going to go drive for a little bit in a straight line and you have an average speed over that time that's really close to what you are. These are fluctuating paths that go forward, you know, forward and backward and side to side and all this kind of stuff. And so their distribution of velocities is actually step by step. So if it's a fluctuating cargo and it takes one step forward and one step back, that counts as a positive velocity and then a negative velocity. Now, if you take the absolute value of it, then you might say, okay, that's a +1 or whatever this speed is and whatever this speed, I have two segments, two time steps at that speed. But of course, I didn't move overall. In fact, you know, I just went forward and backwards. So there's some contribution just of the fluctuations to what might be called these segmental velocities. So that's something to pay attention to. The other thing that they, you know, that they do, well, I'll show you that there's some choices to be made. You could call it a segment velocity. So as soon as you put, excuse me, this point and this point, I believe that's what they did in these in these in these charts. You could call it an average speed over that region. And then, okay, now I have an average velocity. That makes sense. But we'll see in a moment that there's something that even gets a little bit, unsettled there. So let me, first of all, just break down the principles of segmentation analysis. So this is what we're going to try to do in this talk, and this is what I've spent a lot of time with my collaborators thinking about this segmentation analysis and trying to automate things as much as possible. So that's the first thing is, you know, so let's dig into what's called the changepoint detection literature and try to come up with the best possible mechanism that doesn't rely on somebody to segment the trajectories. We want to have you know, like these models can get really complicated if you want to, if you're doing full stochastic differential equation models. So can we build a model that's faithful to the dynamics that are going on, but then is still tractable to analyze? And so I'll talk a little bit about that. But then this last piece, which is one that caught me by surprise, is that how we summarize the data. There are actually different choices that you can make, and you ask yourself, well, which one of these, you know, choices is the one that I should use? Is there a reason to prefer one perspective over another? And what has become clear over the years is that that change point detection will never be perfect. These are intrinsically noisy systems, and depending on the settings and depending on the assumptions that the change point algorithm is built on, they're going to give you different answers. And so what you really don't want to have is a biological analysis that is highly sensitive to the properties of the change point detection algorithm, right? You want to rely on statistics that if I change the change point algorithm, I still reach the same conclusions. So, okay, so let me show you real quick. So that's the backdrop, and let me show you real quick what one of these paths really looks like. So this is a path where what you can see is that it starts, I guess, in the upper left over here, and you know the fact that so this is on an XY. You'll see a lot of these. So there's the X direction and the Y direction, and then we map it so that this is the X time series and the Y time series. So the black is you know, we're following the actual particle locations. And then unfortunately, you know, nature doesn't give us the blue. So what is the blue? We call this the anchor process. So, you know, these motors, you know, the sort of, you know, the just so story, or the thing that would be nice is if there was like one motor that was carrying each cargo. It's not true. There are multiple motors on each cargo. So the first thing to understand is that we're going to infer what we call an anchor process, but it's a phenomenological anchor process. It's saying, suppose that there was this little, you know, little thing that was moving along and the cargo was attached to it on a little rope and it's just fluctuating around it. So the blue that you see here is sort of a best inference from our algorithm about where this anchor might be. You can see that there's fluctuations around it. And the first thing I hope that you take away from this is that at first glance, you might say this particle, just looking at the XY coordinates, starts up here in the upper left, it moves, and then it's stuck, right? What happens when you zoom in on these particles is that you see that these times of being stuck are actually much more active than you think they are. And so, for example, in this one, there's like this definite trend from, I guess that would be from right to left, and then it reverses and goes back left to right. And then there are other periods where it's clearly flat. Now you'll notice that there's this color coded, this green and this red. That's our algorithm again. And this particular color coding is based on just a cutoff of 100 nanometers per second, which we are not claiming is the best way to do it. It's just a way to help the eye in terms of separating, faster and slower segments. So even though these look like blobs, there's actually activity happening underneath. And because you have these microtubules, the dominance goes from one motor to the next. It will move in one direction, but then reverse and come back in the other direction. And sometimes it'll pause in between. And it's really important to emphasize, if you look right here, I hope y'all can see my cursor, that obviously the changepoint algorithm didn't catch this little hitch. So it's going up and then it comes down and then it goes up again. One of the real challenges in this thing is that the algorithm has to deal with the X and the Y axes simultaneously. And it turns out that was an open problem for the kind of data that we're looking at that the graduate students solved, and which was a great breakthrough for our lab. But no matter what, there's just going to be some of these segments that we're going to miss. Okay. All right. So one of our goals in the end is to be able to, how are we going to know, how we know we're doing a good job? One way to do that is to develop, simulations using our model that I'll describe in a little bit. And, by the way, the fluctuations look larger here, but this is because that last one is over a 30-second window. This is over an 8-second window, so it's a little bit more spread out. But I wanted you to be able to see the details. So here, the orange represents, we simulate a real anchor path. The model is straightforward. So we pretend like this anchor is real. You have a sequence of segments that I'll describe the rules for each of them, but there's a velocity and there's a duration associated with each. It moves, and then the cargo fluctuates around it, and then the thing turns again, right? And if you apply our change point detection algorithm and you go through the labeling, you can start to do things like say, okay, the orange is the true anchor location. The blue that I hope you can see is the inferred anchor location. And then, we have periods of stationarity, which are the red, the periods of movement, which is the green, and then the gray areas are the places where there's disagreement. We call this the inference gap, right? So we can measure how well, given a certain kind of data, how well does the whole segmentation analysis do? So, so let me first of all sort of introduce something that caught me by surprise, and we had to pause on it. And I remember we had a slide one year. I was working with Will Hancock, who was at Penn State. He has some data that comes up later. We find ourselves asking, well, what do we mean by a velocity distribution anyway? We have a simulated path here. And so at this time, we were thinking about microtubules that were pretty straight, and it was in vitro transport. So we would project onto a longitudinal direction. So this is the simulated path, and then this is the progress in the longitudinal direction. This is the fluctuations in the transverse direction. And we might say, well, based on this path, what is the velocity distribution along the length of this path? And so for the sort of built-in image trackers, they have a rule where they do velocity I was describing, like step by step. And when you do that, you get this sort of a lot of very sort of slow ones because you have speeds like just sort of fluctuating back and forth. This thing is peaked, it's probably associated with this right here. So this is certainly one way that you can do it, and it's actually pretty common in the engineering literature. Now, another way to do it is through what we said, these segmented velocities. So this seems natural, right? So I clearly have an active and then a slower. So I have two different segments. And so what I might say is, look, I have one motile segment and I have one slow segment. I could take the velocity of each of those, and then that represents one notch here, right? And so what's interesting to look at this is that, okay, that's one segment and one segment, but so that would leave you the impression, I guess you could say, that half of the segments are fast and half of the segments are slow. But you could just as easily represent the same path by a time weighting, right? And so now it's like, oh, well, this was active 2/3 of the time. And then this was, and then it was slow for 1/3 of the time. And so you could have that weighting. And so now it's like, oh, okay, there really are two ways to think about this speed distribution. Should it be segment by segment, or should it be in terms of proportion of time? And this is where the fussiness of the change point algorithm really makes a difference. So just as a thought exercise, well, what happens if I have two different settings for my change point algorithm? I have a less switchy assessment and a more switchy assessment. So I showed you the least switchy possible assessments, and you saw that if I'm thinking about the motile proportion, it's half of the segments, but a third, or sorry, or 2/3 of the time. So if I were to go and say, well, what would happen if I did a switchier assessment, right? So I break this down, but with with the change point algorithm is more inclined to call things new segments. Well, in that case, when you look at this, the motile proportion starts to change. And so in the, let me see if yeah, the switchiness bias. So in the switchy one, the motile proportion of segments suddenly becomes 3/4. So there's a real difference between the less and the more switchy assessment. And this is the kind of thing that you can you can articulate more carefully down the road. But if you look at a time-weighted segment count, right? So I'm looking at, okay, when is it motile and when it's not, and count the seconds that it's in those states, then that actually ends up being a more stable count. And you can think about it like this. If you were able to label every frame as either being in a motile state and in a stationary state, then regardless of whether or not you were breaking those up, if it's a motile segment, if it's 3 segments or if it's 1 segment, as long as that period of being motile is the same, the proportion of time that you spend motile is the same. So that's why that turns out to be robust with respect to the change point. And so we're going to prefer to use assessments that are robust in this sense. Okay. And so if we're going to pick an actual formula, we're going to pick, some, speed distribution that satisfies this property. We ended up calling this thing a cumulative speed allocation. So cumulative because we're going to think about it as, so we don't necessarily want to label things as motile and stationary because who knows what the right cutoff for that would be. So what you do is for every possible speed, you just say, what proportion of time do I spend less than that speed? So it's like a cumulative distribution function, which from probability theory, we know that that's a more robust object than probability densities are. But then it's for all possible speeds. And so it turns out, and I'll come back to this data a little bit later, when you're looking at different types of motors, and you basically what's happening here is you say, okay, my inferred cumulative speed allocation is the amount of time that I spend, and this indicator function is saying that my speed is larger than the inferred velocity for that segment, and then divide by the total time. That, for example, kinesin 1, this is the forward motor, and it's thought of to be very stereotyped. It's the workhorse of the cells. It just goes the same, no matter who's attached, kinesin is going to do what kinesin is going to do. Then its cumulative speed allocation basically says that it has little time spent at these speeds, and then it goes up, and then, so what this is saying is that most of the speeds associated with that motor are, right in this range right here. Whereas dynein is much more, goes along, it stumbles, it goes fast, it goes, I didn't know it actually goes faster than kinesin sometimes. It goes fast, it goes slow, and so you see a much more gradual increase. And then the really interesting thing, the biological thing that comes from these series of papers is that when you get one motor, and this is Will's invention, is scaffolding, that he could control what motors are on, and make sure there's just one or a pair, one kinesin, one dynein, dynactin, BIC-D2, so DDB. Basically he could control that. And when you have the plus N motor and the minus N motor, you get this other curve, which spends much more time at smaller speeds. And so two things to take away is that when you have the opposing motors together, you can see the shift in the speeds. But then what's also interesting is that there are lots of, anybody's familiar with the molecular motor literature, a lot of the literature is based on the idea that there are these like set velocities. It's this velocity, that velocity, that velocity, and you're switching among them. What we see when anytime kinesin and dynein are there together in vivo or even in vitro is that it's a much smoother, there are not these stereotypes, switching between these. It's actually a full distribution. Okay. So Let me talk for just a minute about the mathematical model that I mentioned, some of the details that go into it, and then how that will inform some of the inference that we're going to end up doing. So, the mathematical model, so you can have a highly detailed model where you have like a molecular motor that's doing Poisson stepping, or stepping according to a Poisson process, and then the cargo that's fluctuating, and it's an Ornstein-Uhlenbeck process where it's fluctuating over wherever the anchor is. What we found is that those models, while biophysically sound, there's often not enough data to resolve. There are real identifiability issues. Basically, these things are fluctuating so fast that you can't resolve all of the physical constraints. And so we just think of these things as having these linear segments, and then you just have fluctuations. The cargo, it turns out the fluctuations are very fast with respect to each time step. So if it was lagging behind in the previous step, it might be lagging behind in the next step, or it might be out front. The way to think about it, even though these cargoes are really big, they're fluctuating super fast. And that motor is just sort of like, stepping along. And you can think about this thing. Its main job is not to drag it through the cell. Its job is to hold it and make sure that it's just making steady progress. So that's why we have these like stationary and motile states. And every time you enter into, so the way we modeled it was that we go from one state to the next, and we treated it as a Markov process going from stationary to stationary, stationary to motile, or otherwise, depending on whether you're in a stationary or a motile state. In a stationary state, and by the way, this term stationary has a mathematical meaning. I'm not a huge fan of us using that word, but we prioritize what made sense to the experimentalists, and that word was one that was easier to communicate in the biophysical community. So stationary just means velocity zero, and then you have fluctuations around it. And then if you're motile, the speed is chosen from a gamma distribution, and we had some parameters for that. And then the direction is uniformly distributed 0 to 2 pi. And actually, it got much more tricky than that. In the full paper, we have like forward, and then there's a probability of reversing. And so we do put in some things, but we stop short of trying to put down a microtubule network and having the things move around. Keisha Cook and Veronica Chocanel have been working on something like that, to try to up the realism of these models. So in any case, you have this anchor process, you simulate it. The cargo position then fluctuates around the anchor process, looks something like this. For initial conditions, you have to think about things. So there can be dependence. I was going to say, it's still unclear, but there should be some dependence, and there's some evidence of dependence between the speed and duration of a segment. There's a really easy explanation for that. It's that the microtubules have a finite length, and in fact, they intersect each other. And so the time until you hit an obstacle is obviously affected by your speed. If you're moving really fast, you're going to get there sooner, so your segment's going to be smaller. So there's this inverse relationship between speed and duration. That relationship makes this a non-Markovian process. So you need to do like a burn-in, you pick an initial time and then let things go. We sort of do things in a discrete way. And then when we're thinking about the theory, we're like, okay, let's translate this to the idea that there's a time-dependent state, that's J of t. There's an anchor location, it's a vector, and there's an anchor velocity in any given time. And so what is this object, the cumulative speed allocation, using the ergodic theorem? So phi of s is the cumulative speed allocation. It's literally in stationarity. So if you let the thing move out to time infinity, what's the probability the velocity is less than s? That's what it is. And by the ergodic theorem, that's the same thing as saying if I take one path, and I integrate from time, 0 to t, and here is my typo that I knew was coming at some point. It should be 1 over t times this. So you take the time average, then this is equivalent to this this property here. And so we have different ways to articulate this object, the statistical object that we want to use as our metric. So we, not by coincidence, we've been thinking about things like this is why we chose it. So a few years back, working with Pete Kramer and John Fricks and Veronica Chocanel, we worked on a renewal reward framework for studying asymptotic properties of switching processes like this. And so it turns out that what you can do to study something like this is, you basically you identify what you might call a base case. So in our case, a base case might be the stationary state. So you can be stationary. Now, why are you allowed to go stationary, motile, motile, stationary? Because you might go motile this way and then you might turn. So it's a change in velocity. You can have two consecutive motile states. You could also have a motile state and then you could reverse, right? So there's like different speeds that can happen. So you have like a geometric number of motile states that occur, but then you revisit that stationary state. And so for any property you may be interested in, so that might be the asymptotic diffusivity, or in our case, this cumulative speed allocation, what you want to do is basically say, hey, for each of these cycles, how much, will I accrue in my, object of interest, whether it's square distance or if it's time, proportion of time spent, or I should say time spent less than a particular speed, then I can basically say, whatever that average is. So for the cumulative speed allocation, for example, I could say, how much time do I spend? So sigma is is the length of the stationary state. And then I'm saying add up all the motile states where the velocity is less than s, and then divide that by the expected time of the whole regeneration. And so the point of this 2020 paper is that you can make a calculation like this. You can do even though the true object is like you want to know the expected value of a ratio, it actually lets you commit the sin of computing the average in the numerator and computing the average in the denominator, and it gives you the right answer. So that was the heart of this particular analysis that we did a few years back, and then and in that we were interested in the asymptotic diffusivity of multi-state processes, and it turned out to be exactly the tool we need for this cumulative speed allocation. And so, in our recent, in our submitted work, so we're doing the revision, right now. The most important thing to know here is that, given the kind of process that I have described, there's this interesting constant that depends on the parameters of the system. And then you get that the cumulative speed allocation is this ratio. So F gamma of S, this is actually the CDF, the cumulative distribution function of the speed distribution, where these parameters can be affected by whether or not we assume that speed and duration are dependent. And this thing being a CDF, this is less than 1. So you see this is some number less than 1 that grows to 1 as S goes to infinity, plus this constant rho over 1 plus rho. So it has this nice sort of like, it grows to 1, which is what we expect for an allocation like this. And, just as validation, we have 3 different data sets, and I won't go into all the parameters. I'm happy to talk about it. But basically, we wanted to have what we called our base simulation, which is realistic parameters, but with a clear separation of the motile and stationary states. So the gray that you see here is the lysosome behavior in the I think the cell periphery. So, the base, which has a clear separation in the motile and the stationary states, has a real distinction. So the dark line is the theoretical line that the CSA, that I gave you. And then the orange is if we run these simulations, we run our change point algorithm, we infer the CSA from that, we do a bootstrap. So we basically do bootstrap resampling of the paths. So if there's 200 paths, you compute, for those 200, then you do a bootstrap resampling, you compute the CSA again, you do that again and again, and it gives you a natural degree of variation. We had one called contrast because it's a nice contrast. These are really fast moving motors, and so you can see that they're way out there on the other side. And then we had one called mimic, and what's interesting about the mimic data set is that there is really not a clear separation between the motile and stationary states, which is an interesting lesson. It's what this says is that our protocol tells, this simulation and inference on our own simulations tells us if there is a clear separation between the motile states and the stationary states, we would be able to pick it up. And the fact is that the data is matching the parameter regime where there's not that clear separation. So in that earlier plot, the different, situations I showed you, this is much more like that Ken 1 DDB situation where you have the opposing motors and there's not a stereotype behavior. It's just a clean, just sort of unimodal distribution. Okay. And just to show, to go back and get an idea that this particular method is working, we saw, which something we reported by ad hoc methods in our first paper together. Now we can confirm it with these methods that there is a real difference between the periphery and the paranuclear region. Stuff is moving faster in the periphery. But there's not a significant, so one of the things you fear is that you just have two populations, you always get a difference or something like that. And so these are in the periphery. If you take the large versus the small, there's not a clear, a clean separation between them, which says that you can't reject the null hypothesis that there's no difference between large and small. So we have this nice validation of this. And, part of what I want to say on this front is that the difference when you tease out like segment durations and things like that, what you find out is that in the paranuclear region of these cells, the particles just say they enter the stationary phase and they stay stationary for much longer. So basically they just become bound and then they don't move. And it's not really clear why that happened. And so the graduate student, Nathan Rayans, in his dissertation, took our methodology and basically, entertained a whole bunch of different possibilities, and, did different, genetic disruptions to cells. And what he found was that the key was the endoplasmic reticulum, that they literally took the endoplasmic reticulum, and in some cells, I forgot the word he used, but they dispersed it throughout the cell. And so if the ER is suddenly throughout the cell instead of just right by the nucleus, then sure enough, the lysosomes move like the paranuclear lysosomes in the undisturbed cells. So you really can, this fine, ability to be able to distinguish between populations, but then to also say, nope, some of these are basically the same, allowed him to, basically, make the claim that there's a genuine, like some kind of binding that's occurring that wasn't understood before. So that was good. And he had a cool slide. I meant to include it, but I didn't get it, where he estimated the number of hours that he saved by automation and how many hundreds of cells that he could do, which was really cool because it used to take, hours to do each particle in each cell. So we're really happy to see this, enter into his dissertation in a meaningful way. You can even, so I got about 5 minutes left, so I'm going to go through this part kind of quick, but you could even do things like, one of the first questions that Christine Payne asked me when we started the collaboration, she's like, whenever I go and run these observations, I'm just basically taking a stab in the dark in terms of the frame rate, but I have options. I can do other things. She had been using 1 Hertz data. Now, these simulations that you're seeing here, I think, are from the contrast data set. So they're very extreme in the sense that it's like a big motile state and then it sticks, and then it's a big motile state and then it sticks. So it's not realistic in that sense, but it does show you an interesting thing, which is that if you observe these things at 1 frame per second, you really do sort of see this sort of overall Brownian motion, right? You see the switching between the two. And so mean square displacement maybe is a useful tool in some sense to see what overall movement is. Excuse me. But if you keep the number of observations fixed, which is true of some cameras, and is also true in terms of the computational burden of doing the change point analysis, if you keep the number of observations fixed, what you'll notice is that you get much better information about what's happening in the path, but you also see much less of the phenomenon. You see much less going on. And so there's this fascinating, really, this trade-off that starts to emerge where, if you're trying to, for example, estimate the proportion of time spent motile, at 1 Hertz, you actually make large mistakes because you're averaging over. Like when you're only seeing, when you're just ignoring everything that's happening for a whole second, you're seeing some active and some motile stuff getting mixed together. And so you get really bad estimates for the percentage of time spent motile. And then above a certain point, it becomes, more accurate to the true values. But notice this variance that gets larger. So as the frame rate gets larger, the variance of this estimate gets larger. And then you can you can dig into this thing and look at like false positive, false negatives. That means it's, we know from simulations, is the particle truly moving or not? And so you start to make more false positives when the frame rate gets larger. And that's basically because you could have like a whole short segment where you're just off by a little bit, you get the entire segment wrong. So basically, if you're, you would think, oh, just observe it as fast as you can possibly observe it. It turns out not to be necessarily the best strategy to do that. Let me do this. So, the quick summary of everything we've seen so far is just that that basically we have this tool, we have the statistic that we recommend that people use, maybe as an alternative to mean square displacement, called the cumulative speed allocation. It's sensitive and robust at the same time. You could do things like talk about optimal frame rates. It's led to novel biological observations, and what's really interesting from a functional data analysis point of view is that you can start to think about, well, is this consistent in some sense? Do we have some kind of like theoretical guarantee that we're going to recover the true parameters of a model or something like that? So I want to spend just a little bit of time on that last part with the inference. So with my last 5 minutes, let me just briefly talk to you. So, there's something misleading in the proportion of time spent on different ideas in this talk, in that we probably spent much more time on the change point detection algorithm than we did on the model. This has been, I did not mean to spend 10 years of my life on this, but we did. And it's really hard. This is, it's very difficult to get these things in a way that you feel comfortable. And, I just want to say that, like, in the original work, we use what is called a change in mean detector. So, when we were thinking about it, we were like, okay, well, these things are, they're moving, they're changing their velocities. So let's look at the mean, let's look at the displacement that takes place in every step. And we should see, like, okay, there's like this mean, this displacement, it tends to be, 10 nanometers per step. And then, oh, now it's stopped. It's 0 nanometers per step. So you're looking for changes in the mean displacement. But we notice that what can happen is that if you have a situation where I've got one that's going, one segment clearly moving up, this segment's going down, but there's actually a short, fast segment here. And if you're just looking at, okay, what's the mean displacement here? What's the mean displacement here? These guys right here will get caught up in this sort of negative thing, and you'll just miss it. You just won't see it. On the other hand, if you use what's called, a change in velocity, or this is a generalized, sometimes in 1D, this would be called a change in slope problem. You recognize that this anchor is a continuous process. And so there's no way that you can get from over here to over here without doing something. And so we needed to develop, and the same kind of thing happens here, by the way, where the change in mean will gladly fit this and then have a discontinuous jump. So it was a real technical challenge to go and do a fit that involves continuous piecewise linear anchors. Okay. And so, oops, let me go back up. Oh yeah, I was just saying, you could even, in the change in mean sometimes, because it doesn't pay attention to the order of things, would take this entire segment and treat it as just one, and miss out on sort of these short, fast segments. So that was a real significant problem. Lindo is a student who is a statistics student. One of the nice things here at Tulane is it's a math and stat department, so we attract graduate students who are very statistically adept, which I was not whenever I started out as a professor. I had to go to school on statistics since I've been here, and it's been great. But basically, how do all change point algorithms work? You basically say, well, I think the change points might be here, based on your probability model, which is often, so in this case, it's like I'm going to lay down my anchor and then I'm going to have Gaussian noise around it. Then that's a likelihood. So the log likelihood can be the fit. And then based on the complexity of the model, like if I wanted to have a change point at every single step, I could chase this and have a quote unquote perfect fit, right? But that's not the model, so you have to penalize that. So you have to have that balance between goodness of fit, but also a penalty for complexity. And then you have to go and find the next change point vector. And this actually turns out to be quite a bit of a challenge as well. And so let me see, I got maybe 1 more minute. So I'll just say, what kinds of things make the search difficult? Well, one thing is that if you look at the likelihood landscape for different kinds of trajectories, the maximum likelihood is here, but there are going to be local maxima. And so this is a case where there's just 2 changes, But you can imagine a situation you have like 10 or 12 changes. You're going to have all kinds of little maxima all over the place. The other problem is that when you have this is specific to the change in velocity problem, not the change in mean, not the change, the change in mean one doesn't have this problem. You can't just go and say, well, let me find the best fit when there's one change and then keep that and then go up to the next step and say, add in a new one. So if this was a change in means one, you would get, oh, okay, well, if I'm going to add one, this is a good place to put it. But if you look at the score, so this is like, suppose I put the change point wherever, and here be dragons, right? So, here, if this is the score, if I place the change point at any of these places. So if I'm going to pick a place to put a change point to give the best possible fit, it's actually going to end up being right in the middle. So it's going to be neither of the correct answers. And so any search that involves saying, let me fit one, okay, that's good. Now let me fit two, or let me fit three, is going to fail immediately. Similarly, there can be valleys. If you want to go here from, say I have a fit when, there's 0 changes, then clearly there are 2. It turns out that picking either of the other 2, either one of the correct changes has a lower score than 0 or 2. So basically, if you're always making one-step moves, you're not going to be able to get across valleys. And so we had to develop a random walk in change point space. And then do it in such a way that it's it was a reversible process so that we could, do Metropolis Hastings post proposals and then evaluations that we could ultimately make an MCMC. We basically do an MCMC walk through change point state and then try to find the best, after we do 10,000 steps of a walk, we go back and look and say, okay, what was the best fit that we came across? And then we declare that's the stochastic search part of it. And so, I'm out of time, but basically, we have the fit, we have the search, and then there are 2 pieces that are very technical in terms of information criteria, and we had to put a speed penalty so it doesn't chase noise. And Lynn was actually able to prove a theorem, the consistency theorem, based on empirical process theory developed by Talleyrand and then brought to statistics by Sarah Vandegeer. So it's a really nice piece of theoretical work that she did, and she'll be defending that later this month. So let me just skip to the to the end and basically say, where are we going from here? So we have this nice tool where we can sort of we have this whole allocation, but ultimately we need to think about the stationary motile dichotomy. When we see sort of a long, slow segment, or we see something that's a very small velocity, can we say that can we reject the fact that it is actually 0? Or, can we say, well, that's indistinct, that the thing is within the realm of natural variation, and so it may be stationary and it moved. So anyway, so that's where we're sort of going, and that's a chapter of our dissertation, and we hope to make some progress on that this summer. So thank you very much for your attention. I'll take any questions that y'all have.
Okay. Thank you. Any question